Part of my job is actually talking to people about spatial transcriptomics, but one thing that makes it kind of tricky is a lot of people really haven't heard of it yet. So I often catch myself trying to explain it from scratch. I look for the easiest ways to describe it while still trying to highlight just how incredible it actually is. Hey, it's me, Bruno. Welcome back to Bruno's BioLogs, a space where we explore the frontiers of the life sciences together. And today, I want to take you on a quick journey into the world of spatial, or as I like to call it, turning gene expressions into a living map. I live here in San Francisco, and imagine if I just gave you a spreadsheet with all the city zip codes and expected you to understand a place with just that. Sure, you would know which neighborhoods exist, but you wouldn't really know what they look like. You wouldn't get the vibe, the culture, where the best coffee shops are, or that walking from the Mission to Noe Valley might actually destroy your calves. Like seriously, these hills do not play around, they will destroy your calves. And for the longest time, that's exactly what studying gene expression was like. We knew which genes were active, but we had no idea where those cells were in the tissue or what they were surrounded by. Then spatial transcriptomics came along and flipped on the city's lights. Suddenly, we weren't just seeing what genes were turned on, we were seeing where they were active and who they were interacting with. We weren't looking at spreadsheets anymore, we were actually walking through biological streets. So, where did this all start? Well, back in 2016, a team in Sweden had this clever idea. They took standard microscope slides and printed them with tiny little spots, each with unique DNA sequence, kind of like a molecular name tag. Then when a tissue sample was placed on top, the RNA floating out of the tissue would land on these tiny little spots and pick up the tag, like picking up a postal code. So later, when we sequenced the RNA, we didn't just know what gene it came from, we knew exactly where in the tissue it came from. And just like that, we had a map. And from there, the field has really taken off. There are tons of different approaches now. Some using slides, others using beads, some zooming in super close, and others just covering entire organs. They all ask the same questions. Where are the genes and who are they hanging out with? So why does this actually matter? Say we're studying cancer. Spatial maps can actually show us whether immune cells are sneaking into the tumor or they're getting blocked at the edge. In brain research, we can actually figure out which neurons are expressing memory-related genes and where they are in the brain. In developmental biology, we can actually trace how organs form from a flat sheet of cells into complex 3D structures. It's kind of like upgrading from a crumpled tourist map to Google Street Views, but for your cells. And I bet you're wondering, oh Bruno, what's your favorite part? Spatial transcriptomics doesn't try to replace these older methods. It actually builds on top of them. Just like how Google Maps didn't try to replace street names, it just gave you context, layers, and direction. Traditional techniques were just telling us what was happening, and spatial just adds the where. And when you know what's going on and where it's happening, suddenly biology just starts making a lot more sense. So what's next? We're starting to stack even more info, combining RNA, protein, DNA structures on the same tissue. We're building 3D maps of entire organs. Some folks are even trying to watch these changes unfold in real time. So yeah, spatial isn't just this fringe tool anymore. It's becoming central to how we study health, disease, and development. Look, I don't study every gene expressions out there. I really don't claim to. But the one I do work with, I think it's pretty incredible. And hopefully, after watching this, you're at least a little curious too. Want to talk more about spatial? Shoot me a message. I'm always down to nerd out about it. From spreadsheets to spatial maps, biology has never looked more alive. Thank you so much for watching. Hit me up. Let's connect. I'll see you the next time on Bruno's BioLogs.